Tonight's lecture is over Chapter 3, Molecules of Life. So for this chapter, our learning objectives are to describe the form and the function of each of the four macromolecules, to describe the different categories of carbohydrates and their uses, to compare and contrast different categories of lipids and their structures, to understand the importance of the shape in the functioning of a protein, and to compare and contrast DNA and RNA. A little bit of chemistry goes a long way in the study of biology and can help in the understanding of a great deal about your everyday life. Will eating those beans in your soup keep you up all night with gastrointestinal distress? Just knowing whether beans and lentils are lima beans, each contains slightly different types of sugar molecules, will give you an answer. For example, will the butter you spread on your toast sabotage your efforts to lose weight? Understanding something about the carbohydrogen connections in the fat molecules can help you figure out the answer. The foods in our diet would seem to have nothing in common with a daisy or a chinchilla, or a bacteria. Yet all are made from organic molecules, that is, chemical compounds that contain both carbon and usually hydrogen. In this chapter, we looked at tiny hydrogen with its single proton and electron, as well as simple molecules such as water. In this chapter, we'll look at much larger molecules. Because many of the organic molecules important to life are made up of similar building blocks and may have thousands of atoms in a single molecule, they are called macromolecules. 4 types of macromolecules are essential to the building and functioning of living organisms, and they are carbohydrates, lipids, proteins, and nucleic acids. All organisms on Earth largely consist of these types of molecules. Furthermore, there is a tremendous variety among the 4 primary macromolecules that make up all organisms' structures and chemical machinery. But a common theme emerges when we look at many of these macromolecules. Relatively simple sets of building blocks called monomers are linked together by covalent bonds. From this simple process, it is possible to form an almost infinite variety of complex molecules called polymers. We will see that among carbohydrates, polysaccharides such as starches are polymers of simple sugar monomers. Proteins are polymers made up of the 20 amino acids, and the nucleic acids, RNA and DNA, are polymers constructed from sequences of the 4 different nucleotide monomers. On the other hand, lipids, which are defined by their chemical properties of insolubility in water, are not constructed as polymers. And that brings us to our take-home message for section 3.1, which is macromolecules are polymers of large organic molecules made up from smaller building blocks. And there are 4 types of macromolecules that are essential to living organisms. And those 4 macromolecules that you need to be familiar with are carbohydrates, lipids, proteins, and nucleic acids. Moving on, carbohydrates are the primary fuel for cells and also for much of the structure of cells in all organisms. Carbohydrates always contain carbon, hydrogen, and oxygen, and they sometimes contain atoms of other elements. A carbohydrate has approximately the same number of carbon atoms that it does water units in its chemical formula. Carbohydrates are classified into several categories based on their size and their composition. The simplest carbohydrates are monosaccharides or simple sugars, which contain anywhere from 3 to 6 carbon atoms. When they are broken down, they produce molecules that usually are not carbohydrates. 2 common monosaccharides are glucose, found in the sap and fruit of many plants, and fructose, found primarily in fruits and vegetables, as well as in honey. Fructose is the sweetest of all naturally occurring sugars. The suffix OSE tells us that a substance is a carbohydrate. Although sugars have the same composition of atoms and chemical formula, the arrangement of the atoms differs slightly, giving them different characteristics. Carbohydrates function well as fuels because of their many carbon-hydrogen bonds. As those bonds are broken down and other, more stable bonds, primarily between carbon and oxygen, are formed, a great deal of energy is released that organisms can use. The most important carbohydrate for living organisms is glucose. This is a monosaccharide usually found in fruits. Most of the carbohydrates that you eat, including table sugar called sucrose, and the starchy carbohydrates found in bread and potatoes, are converted into glucose in your digestive system. The glucose then circulates in your blood at a concentration of about 0.1%. Circulating glucose, also called quote-unquote blood sugar, has one of three fates. Firstly, it enters the cell and is used for fuel for cellular activity. The process that releases energy from the high-energy bonds of glucose is discussed in detail in following chapters. It is also stored temporarily as glycogen. If glucose is circulating in your bloodstream in excess of your body's needs, it can be stored temporarily in various tissues, primarily your muscles and liver. The stored glucose molecules are linked together to form a large web of molecules called glycogen. When you need energy later, this glycogen can be easily broken down to release glucose molecules back into your bloodstream. Glycogen is the primary form of short-term energy storage in animals. That brings us to water weight. Glycogen also plays a role in the relatively rapid weight loss that you might experience if you're dieting. Glycogen stores in your muscles and liver are the first, most accessible molecules that can be broken down for energy in the absence of sufficient sugar in your bloodstream. And large amounts of water are bound to glycogen. Consequently, as that glycogen is removed from your tissue, so too is the water. This loss of water is responsible for much of the initial weight loss that occurs before your body resorts to using stored fat. Then the rate of weight loss slows considerably. Thirdly, glucose circulating in your bloodstream can be converted into fat, a form of long-term energy storage. And that brings us to our next take-home message for section 3.2, and that is carbohydrates are the primary fuel for cells and also form much of the cell structure. Glucose is the most important carbohydrate for living organisms. Also, glucose in the bloodstream can be used as an energy source. It also can be stored as glycogen in the muscles and the liver for later use, or it can also be converted into fat. Moving on, in contrast to the simple sugars, some carbohydrates contain more than one sugar unit or building block. That is, they are polymers of simple sugars bound together. A disaccharide forms when two simple sugars bond together. The joining of glucose and galactose, for example, makes the disaccharide lactose, which is the sugar found in milk. When large numbers of simple sugars are joined together, the resulting carbohydrate is called a polysaccharide or complex carbohydrate. Glycogen consists of about 30,000 linked glucose molecules. Depending on how the simple sugars are bonded together, polysaccharides may function as time-release stores of energy or as structural materials for plants that may be completely indigestible by most animals. Many disaccharides and polysaccharides are important sources of fuel for cells, but they must undergo processing before their energy can be released from their bonds. Moving on, let's look at what happens when we eat sucrose. This is common table sugar. Sucrose is the primary carbohydrate in many plant-based foods and is composed of the linked monosaccharides glucose and fructose. To use sucrose, the body first breaks the bond linking the glucose and the fructose. The individual monosaccharides are then broken down into their component atoms, and the energy that was stored in their chemical bonds is harvested and used. In plants, the primary form of energy storage is a complex carbohydrate called starch, and this is found in the roots and other tissues. Starch consists of linked glucose molecules. Grains such as barley, wheat, and rye are high in starch content, as are corn and rice, both of which are more than 70% starch. Many complex carbohydrates, particularly starches that have been highly processed, are like time-released fuel pellets. The glucose molecules become available slowly as the bonds between glucose units are broken. In contrast, many refined sugars and refined starches give a quick burst of energy because the sugars are more immediately available. And that brings us to our next take-home message for section 3.3, and that is complex carbohydrates composed of linked simple carbohydrates include starch, which is the primary form of energy storage in plants, and glycogen, which is an important form of energy storage in animals. Moving on, despite their importance as a fuel source for humans, not all carbohydrates can be broken down by our digestive system. Two different complex carbohydrates, both indigestible by humans, serve as structural materials for invertebrate animals and plants, respectively, chitin and cellulose. Chitin forms the rigid outer skeleton of most insects and crustaceans. In contrast, cellulose forms a large variety of plant structures and is found in trees, cotton, and leaves and grasses. In fact, because all plants have cellulose as a component of their cell walls, it is the single most abundant compound on Earth. Surprisingly, cellulose is almost identical in composition to starch, but one small difference in the chemical bond between the simple sugar units changes the three-dimensional structure of the macromolecule. Even tiny differences in the shape of a molecule can affect its behavior. In this case, the difference in shape makes cellulose a sturdy structural material that is impossible for humans to digest. Although it is not distinguishable, cellulose is still important to our health. The cellulose in our diet, known as fiber, is also appropriately called roughage because as the cellulose of celery stalks, lettuce leaves, and the like passes through our digestive system, it scrapes the wall of the digestive tract. Its bulk and the scraping stimulate the more rapid passage of food and of unwanted, possibly harmful products of digestion through our intestines. In this way, fiber reduces the risk of colon cancer and other diseases. But because it reduces the time for water reabsorption in our intestines, too much fiber can lead to diarrhea. Unlike humans, termites and ruminant mammals, such as cows, have microorganisms living in their guts that are able to break down cellulose. That's how termites can chew on wood, and ruminants can extract usable energy from the freed glucose molecules of cellulose in the grass and other plants that they eat. And that brings us to our take-home message for section 3.4 of our book, and that is some complex carbohydrates, including chitin and cellulose, cannot be digested by most animals. Indigestible carbohydrates in the diet, called fiber, aid in digestion and have many health benefits for humans. Moving on, lipids are a second group of macromolecules important to all living organisms. Lipids, like carbohydrates, are made primarily of atoms of carbon, hydrogen, and oxygen, but the atoms are present in different proportions. The lipids in your diet, for example, tend to have significantly more energy-rich carbon-hydrogen bonds than do carbohydrates, so they also contain significantly more stored energy. Lipids do not dissolve in water and are greasy to the touch. Think of butter or olive oil. Lipids are insoluble in water because they tend to have long chains consisting only of carbon and hydrogen atoms that are nonpolar. This means that there are no regions of positive or negative charge, so they do not form weak hydrogen bonds with water and cannot dissolve in water. Nonpolar molecules tend to minimize their contact with water and are considered hydrophobic, or what we call water-fearing. Lipids cluster together when mixed with water, never fully dissolving. Molecules that readily form hydrogen bonds with water are called hydrophilic or water-loving. One familiar type of lipid is fat, the type most important in long-term energy storage and insulation. Penguins and walruses can maintain relatively high body temperatures despite living in very cold habitats due to their thick layer of insulating fat. Lipids also include sterols, a group of organic molecules that includes cholesterol and many of the sex hormones that play regulatory roles in animals, and phospholipids, which form the membranes of cells. The chemical breakdown of fat molecules releases significantly more energy than does the breakdown of carbohydrates. A single gram of carbohydrate stores about 4 calories of energy, while the same amount of fat stores about 9 calories. A calorie is the energy needed to raise the temperature of 1 gram of water by 1 degree Celsius. The difference in calorie storage within carbohydrates versus fat is similar to the difference in currency storage between $5 bills and $10 bills. Because fats store such a large amount of energy, a strong taste preference for fats over other energy sources has evolved in animals. This preference helped the earlier humans to survive. Today, however, it puts us in danger from the health risks of obesity when we overconsume fats. And that brings us to our take-home message for section 3.5, and that is lipids are insoluble in water and greasy to the touch. They are valuable to organisms for long-term energy storage and insulation, in membrane formation, and as hormones. The high calorie content per gram of fats is why humans prefer fats to other macromolecules in the diet. Moving on, all fats have a quote-unquote head region and 3 long quote-unquote tails. The head region is a small molecule called glycerol. It is linked to tail molecules known as fatty acids. A fatty acid is simply a long hydrocarbon chain. That is, a chain of carbon atoms, often a dozen or more, linked together and with 1 or 2 hydrogen atoms attached to each carbon atom. The fats in most foods we eat are what we call triglycerides, which are fats having 3 fatty acids linked to a glycerol molecule. For this reason, the terms fats and triglycerides are often used interchangeably. Triglycerides that are solid at room temperature are generally categorized as fats, while those that are liquid at room temperature are classified as oils. The terms saturated and unsaturated refer to the hydrocarbon chain of fatty acids. If each carbon atom in the hydrocarbon chain of fatty acids is bonded to 2 hydrogen atoms, the fat molecule carries the maximum number of hydrogen atoms and is said to be a saturated fat. When saturated, the hydrocarbon tails of the fatty acids all line up very straight, allowing the fat molecules to pack together tightly. The tight packing causes the fats, such as butter, to be solid at room temperature. Most animal fats, including those found in meat and eggs, are saturated. Relative to other animals, however, fish tend to have less saturated fat. Saturated fats are not essential to good health, and because they accumulate in your bloodstream and can narrow the blood vessels, they can contribute to heart disease and strokes. On the other hand, an unsaturated fat is one in which some of the carbon atoms are bound to only a single hydrogen atom, and pairs of neighboring carbons are connected to each other by a double bond. When unsaturated, the fatty acids have kinks in the hydrocarbon tails, and the fat molecules cannot pack together so tightly. Consequently, unsaturated fats do not solidify as easily and are liquid at room temperature. Most plant fats are unsaturated. Unsaturated fats may be monounsaturated or polyunsaturated. A monounsaturated fatty acid hydrocarbon chain has only one pair of carbon atoms in the unsaturated state. That is, it has only one double bond. A polyunsaturated fatty acid hydrocarbon chain has more than one pair of carbons in an unsaturated state. There's more than one double bond. Unsaturated fats are high in calories, but because they may lower cholesterol, they are generally more healthful than saturated fats are. Food high in unsaturated fats include avocados, peanuts, and olive oil. Moving on to trans fats, the ingredient list for some snack foods includes particularly hydrogenated vegetable oils. The hydrogenation of an oil means that hydrogen atoms have been added to a liquid, unsaturated fat so that it becomes more saturated. Unfortunately, hydrogenation also makes food less healthful because it also creates what we call trans fats, with the word quote-unquote trans referring to the unusual orientation of some or all of the double bonds that remain following the addition of the hydrogen atoms. Because of these bonds, the trans fats are less reactive. Your body is less likely to break down them, and so more likely to accumulate in your blood vessels. And that brings us to our take-home message for 3.6. That is, fats, including the triglycerides common in the foods we eat, are one type of lipid. They are characterized by long hydrocarbon tails, and fats store energy effectively in their many carbon-hydrogen and carbon-carbon bonds. Moving on, not all lipids are fats, nor do lipids necessarily function in energy storage. A second group of lipids, called the sterols, are organic molecules with a variety of functions, including important roles in regulating growth and development. This group includes cholesterol and the steroid hormones such as testosterone and estrogen. All of these molecules are variations on one basic structure formed from 4 interlinked rings of carbon atoms. Moving on, cholesterol is an important component of most cell membranes. For this reason, it is an essential molecule for living organisms. Cells in our liver produce almost 90% of the circulating cholesterol by transforming the saturated fats in our diet. With excess cholesterol and fat, a substance called plaque, which is a mixture of cholesterol, fats, calcium, and clotting material, forms, and this can cause hardening of the arteries and contributes to heart attack or stroke. Moving on, estrogen and testosterone are built through slight chemical modifications of cholesterol. These steroid hormones are among the primary molecules that direct and regulate sexual development, maturation, and sperm and egg production. In both males and females, estrogen influences memory and mood, among other traits. Testosterone has numerous effects, one of which is to stimulate muscle growth. Some athletes, particularly bodybuilders, take synthetic variants of testosterone to increase their muscularity and strength. But the use of these supplements may cause dangerous side effects, including extreme aggression, known as roid rage, high cholesterol, and following long-term use, such as cancer. And, as a consequence, nearly all athletic organizations have banned their use. Moving on, phospholipids and waxes are also lipids. Phospholipids are the major component of the membrane that surrounds the contents of a cell and controls the flow of chemicals into and out of the cell. They have a structure similar to fats, but with two differences. They contain a phosphorus atom, and they have two fatty acid chains rather than three. Moving on, waxes resemble fats, but have only one long chain fatty acid linked to the glycerol head of the molecule. Because the fatty acid chain is highly nonpolar, waxes are strongly hydrophobic. That is, these molecules repel water. Their water resistance accounts for their presence as a natural coating on the surface of many plants and in the outer coverings of many insects. In both cases, the waxes prevent the loss of water, and this is essential to the organism's life processes. Many birds, too, have a waxy coating on their wings, keeping them from becoming waterlogged when they get wet. And that brings us to our take-home message for section 3.8, and that is cholesterol and phospholipids are lipids that are not fats. Both are important components in cell membranes. Cholesterol also serves as a precursor to steroid hormones, which are important regulators of growth and development. Moving on, I stand before you hundreds of thousands of proteins. You can't look at an animal and not see proteins. Proteins make up skin and feathers and horns. They make up muscles and are a significant component of bone. In your bloodstream, proteins fight invading microorganisms and stop you from bleeding to death from a shaving cut. Proteins control the levels of sugar and other chemicals in your bloodstream and carry oxygen from one place in your body to another. And in just about every cell and every living organism, proteins called enzymes initiate and assist every chemical reaction that occurs. Although proteins perform several very different types of functions, all are built in the same way and from the same raw materials in all organisms. Just as in the English language, every word is formed from one or more of the 26 letters of the alphabet, so proteins are constructed from a sort of similar alphabet known as amino acids. Moving on, unique combinations of 20 amino acid monomers are strung together, like beads on a string, and the resulting protein is a polymer with a unique structure and chemical behavior. Amino acids contain the familiar atoms as carbohydrates and lipids, carbon, hydrogen, and oxygen, but they also contain nitrogen. At the center of every amino acid is a carbon atom with its 4 covalent bonds. One bond attaches the carbon to a group of atoms called a carboxyl group, which is a carbon atom bonded to 2 oxygen atoms. The second bond attaches the central carbon to a single hydrogen atom. The third bond attaches the central carbon atom to an amino group, which is a nitrogen atom bonded to hydrogen atoms, usually 2 or 3. These components are the foundation that identifies a molecule as an amino acid. As multiple amino acids are joined together, the unit formed by these 3 components forms the quote-unquote backbone of the protein. The fourth bond of the central carbon atom attaches to a side chain. This side chain is the unique part of each of the 20 amino acids, determining that amino acid's chemical properties. In the simplest amino acid, glycine, the side chain is simply a hydrogen atom. In other amino acids, the side chain is a single methyl group, or 3 or 4 such groups. Most of the side chains include both hydrogen and carbon atoms and a few nitrogen or sulfur atoms. Moving on, the atoms present in the proteins we eat, especially nitrogen atoms, are essential to the growth, repair, and replacement processes that take place in our bodies. When we digest protein, breaking it down into its amino acids, our bodies use these amino acids for various building projects. Proteins also store energy in their bonds and, like carbohydrates and lipids, can be used to fuel living processes. Moving on, the amount of protein we need depends on the extent of the building projects underway. Most individuals need 40 to 80 grams of protein per day. Athletes engaging in strength training, however, may need 150 grams or more a day to achieve the extensive muscle growth stimulated by their training. Similarly, the needs of pregnant and nursing women are approximately 15 to 20 grams more per day. All proteins are not created equal. Each protein has a different composition of amino acids, and while our bodies can manufacture certain amino acids as they are needed, most adults typically must get 9 of the 20 amino acids from their diet, and that's why these are called essential amino acids. Food that contain complete proteins deliver all of the essential amino acids. Animal products such as milk, eggs, fish, chicken, and beef tend to provide complete proteins, as do some vegan products such as quinoa and soy. Vegetables, fruits, and grains often contain incomplete proteins which do not have all of the essential amino acids. 2 incomplete proteins that are complementary proteins, when eaten together, can provide all the essential amino acids. Traditional dishes in many cultures often include such pairings. For example, corn and beans in Mexico and rice and lentils in India. And that brings us to our take-home message for section 3.9, and that is proteins are made with unique combinations of 20 amino acids and perform a myriad of functions necessary for growth, repair, and replacement of tissue and living organisms. 9 of the 20 amino acids cannot be synthesized by the body and so must be consumed in the diet. Moving on, proteins are formed by linking individual amino acids together with the peptide bond, in which the amino group of one amino acid is bonded to the carboxyl group of another. 2 amino acids joined together form a dipeptide, and several amino acids joined together form a polypeptide. The sequence of amino acids in the polypeptide chain is called the primary structure of the protein and can be compared to the sequence of letters that spells a specific word. And you can reference your book, page 63, figure 3.17, for this particular cartoon. Amino acids in a polypeptide chain don't remain in a simple straight line like beads on a string, however. The chain folds as side chains come together and hydrogen bonds form between various atoms in the chain. The 2 most common patterns of hydrogen bonding between amino acids cause a segment of the chain to either twist in a corkscrew-like shape or form a zigzag folding pattern. The distribution of corkscrews and zigzags within a protein gives a protein its secondary structure. The secondary structure itself continues to fold and bend, bringing together amino acids that then form bonds such as hydrogen bonds or covalent sulfur-to-sulfur bonds. Eventually, the protein folds into a unique and complex three-dimensional shape called its tertiary structure. Some protein molecules are made from more than one polypeptide chain. The polypeptide chains are held together by hydrogen bonds and bonds between amino acids from different chains, giving the protein a quaternary structure. An example of a protein with quaternary structure is hemoglobin, the protein molecule that carries oxygen from the lungs to the cells where it is needed. Moving on, a protein structure relates directly to its function. Thus, for proteins to function properly, they must retain their three-dimensional shapes. This shape determines which molecules the protein can interact with. When their shapes are deformed, they usually lose their ability to function. We can see proteins deforming when we fry an egg. The heat breaks the hydrogen bonds that give the proteins their natural shape. The proteins in the clear egg white unfold, losing their secondary and tertiary structures. This disruption of protein folding is called denaturation. Moving on, hair is another protein whose shape many of us have modified at one time or another. Styling hair, whether curling or straightening it, involves altering some of the hydrogen bonds between the amino acids that make up the hair proteins, changing its tertiary structure. When your hair gets wet, the water can disrupt some of the hydrogen bonds, causing some amino acids in the protein to form hydrogen bonds with water molecules instead. Thus, if you style your hair while it's wet, you can change its shape, making it straighter, or if you manipulate it around curlers, making it curlier, the hair holds this shape when it dries because as the water evaporates, the hydrogen bonds to the water are replaced by hydrogen bonds to the amino acids of the hair protein. Once your hair gets wet again, however, unless it is combed, brushed, or wrapped in a different style, it will return to its natural shape. So whether your hair is straight or curly or somewhere in between depends on your hair protein's amino acid sequence and the three-dimensional shape it confers. This amino acid sequence is something you're born with. It's genetically determined. The chains are more or less controlled depending on the extent of the covalent and hydrogen bonding between different parts of the coil. Hair salons use chemicals to alter covalent bonds, which changes the hair texture semi-permanently. And that brings us to the take-home message for section 3.10. A particular amino acid sequence determines how a protein folds into a particular three-dimensional shape. This shape determines many of the protein's properties, including which molecules it will interact with. When a protein's shape is deformed, the protein usually loses its ability to function. Moving on, protein shape is particularly critical for enzymes, that is, molecules that help initiate and accelerate the chemical reactions in our bodies. When reactions are complete, enzymes are unchanged. They remain in their original form and thus can be used again and again. So think of an enzyme as a piece of popcorn. Its tertiary or quaternary structure gives it a complex shape with lots of nooks and crannies. Within one of those nooks is a small area called the active site. Based on the chemical properties of the atoms lining this nook, the active site provides a place for the participants in a chemical reaction, the reactants or substrate molecules, to nestle briefly while the reaction is ongoing. Furthermore, only the substrate molecules are of the correct size and shape to fit in the active site. Therefore, enzymes bond only with their appropriate substrate molecules. Moreover, the exposed atoms in the active site are positioned so that they can engage in weak interactions with specific atoms in the substrates. Once a substrate molecule is bound to the active site, a reaction can take place and usually does so very quickly, after which the altered substrate is released. Moving on, the chemical reactions that occur in organisms can either release energy or consume energy. In either case, a certain minimum energy or a little quote-unquote push is needed to get the reaction going. This push is called the activation energy. Although enzymes don't alter the amount of energy released by a reaction, they do act as catalysts, initiating and speeding up the reaction by lowering the activation energy. For example, enzymes may lower the activation energy by holding substrate molecules in an orientation that stresses bonds that need to break or by bringing together atoms that need to bond. By virtue of their catalytic capabilities, enzymes are at the heart of the chemistry of living organisms. Taken together, all of the chemical reactions in a living organism are its metabolism. And I would just like to interject here that Professor Fugate's definition of death is the loss of the ability to metabolize. And that brings us to our take-home message for section 3.11, and that is enzymes are proteins that help initiate and speed up chemical reactions. The enzymes aren't permanently altered in the process and can be reused. Moving on, if not for enzymes, almost nothing would get done in cells. Enzymes do not alter the outcome of reactions, but without the chemical nudge they supply, often increasing reaction rates to millions of times the uncatalyzed rate, the processes necessary to sustain life could not occur. The rate at which an enzyme catalyzes a reaction is influenced by several chemical and physical factors. And we're going to outline these chemical and physical factors right now. Firstly, there's the enzyme and substrate concentration. The rate of a typical reaction increases with the addition of either more enzyme or more substrate, when an addition causes a higher rate of collision between enzyme and substrate molecules. Secondly is temperature. Because molecules move faster as temperature increases, reaction rates generally increase as temperatures rise. However, reaction rates continue to increase only up to the optimum temperature for an enzyme. At temperatures above the optimum, reaction rates decrease as enzymes lose their shape. Thirdly is pH. As with temperature, enzymes have an optimum pH. Above or below this pH, excess hydrogen or hydroxide ions interact with amino acid side chains in the active site or elsewhere. These interactions disrupt enzyme function and sometimes enzyme structure, and it can decrease reaction rates. Fourth is the presence of inhibitors. Inhibitors, which reduce enzyme activity, come in two types. Competitive inhibitors bind to the active site, blocking substrate molecules from the site and thus from taking part in the reaction. Non-competitive inhibitors, on the other hand, do not compete for the active site, but rather bind to another part of the enzyme, altering its shape in a way that changes the structure of the active site, thus reducing or blocking its ability to bind with substrate. Within a metabolic pathway, often it is the very product of one of the chemical reactions that inhibits enzymes earlier in the pathway, effectively shutting off the pathway when sufficient end product has been produced. And fifthly, the presence of activators. Just as a molecule can bind to an enzyme and inhibit the enzyme's activity, so can some cellular chemicals act as activators. These chemicals alter the enzyme's shape or structure so that it can catalyze a reaction. Moving on, this is figure 3.21 on page 66 of your book. And you can see that this enzyme and substrate concentration is outlined in this graph. The rate of a reaction typically increases with the addition of either more enzyme or more substrate. When such an addition causes a higher rate of collisions between enzyme and substrate molecules. So we had talked about that a few minutes ago. And moving on, this is the same figure, the same page, and now we see how temperature can act on an enzyme. So we see that the reaction rate generally increases at higher temperatures, but only up to the optimal temperature for an enzyme. And moving on once again, we see how pH can aid or reduce the reaction rate of an enzyme. So the reaction rates generally increase as pH nears the optimal level for an enzyme. Looks like a nice bell curve, doesn't it? And then that brings us to our fourth and fifth point in our discussion of enzymes, which is inhibitors and activators. And we can see how inhibitors and activators act on an enzyme with this graph, or I should say these graphs. So we see the reaction rates decrease in the presence of inhibitors and increase in the presence of activators. Moving on, in any case, enzyme functioning is critically dependent on the enzyme's precise shape, which follows from its amino acid sequence. But sometimes a cell produces a protein quote-unquote word that is misspelled. That is, the sequence of amino acids is incorrect. If the structure of an enzyme is altered even slightly, the active site or some other region of the protein molecule may change so that the enzyme will no longer function. Non-functioning enzymes are responsible for a large number of diseases and physiological problems. One example is lactose intolerance. In individuals who can consume dairy products during digestion, the lactose in milk is broken down into glucose and galactose. These simple sugars are then used for energy. But some people, when they are adults, are unable to break the bond linking the two simple sugars because they no longer produce the enzyme lactase, which assists in this process. Consequently, any lactose in the diet of such lactose-intolerant individuals passes through their stomachs and small intestines undigested. Then, when it reaches the large intestine, the bacteria living there consume the lactose. The problem is that, as the bacteria break down the lactose, they produce carbon dioxide and other gases, causing discomfort. Interestingly, in regions of the world with long traditions of pastoralism, that is, raising and consuming livestock, lactose intolerance is much less common than in other parts of the world. Only about 10% of people from Denmark or Sweden have lactose intolerance, but among people from China, which has historically been largely non-pastoral, the vast majority of adults, more than 80% according to numerous published studies, are lactose intolerant. And that brings us to our take-home message for section 3.12. Enzyme activity is influenced by physical factors such as temperature and pH, as well as by chemical factors including enzyme and substrate concentrations. Inhibitors and activators are chemicals that bind to enzymes and can change the rate at which the enzyme catalyzes reactions. Moving on, the fourth of life's macromolecules are nucleic acids. There are two types of nucleic acids, deoxyribonucleic acid, in other words DNA, and ribonucleic acid, in other words RNA. Both play central roles in directing the production of proteins in living organisms and, by doing so, play a central role in determining all of the inherited characteristics of an individual. Moving on, consistent with the common theme we have noticed that characterizes the construction of many macromolecules, nucleic acids are made up of individual units called nucleotides. All nucleotides have three components: a molecule of sugar, a phosphate group, which contains a phosphorus atom bound to 4 oxygen atoms, and a nitrogen-containing molecule. In both types of nucleic acids, the nucleotide monomers are linked in a series. A sugar molecule is attached to a phosphate group, which is attached to another sugar, which is attached to another phosphate, and so on. The linked series forms the backbone of the nucleic acid molecule. Attached to each sugar and protruding from the backbone is one of the nitrogen-containing molecules, the nucleotide bases, so named because of their chemical structure. A 10-unit nucleic acid strand, therefore, would have 10 bases, one attached to each sugar within the sugar-phosphate backbone, but the base attached to each sugar is not always the same. It can be one of several different bases. For this reason, a nucleic acid is often described by its sequence of bases. Moving on, nucleic acids store information in the order of bases attached at each position in the molecule's backbone. At each position in a molecule of DNA, for example, the bases can be any one of 4 possibilities: adenine, or A, thymine, or T, guanine, G, and cytosine, C. Just as the meaning of a sentence is determined by which letters are strung together, the information in a segment of DNA is determined by its sequence of bases. One DNA segment may have the sequence adenine, 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 guanine, cytosine, thymine, guanine, cytosine, thymine, abbreviated as A, G, C, T, G, C, T. Another DNA segment may have the sequence CGATTACCCAT. Because the information differs in each case, so too does the polypeptide for which the sequence codes. And that brings us to our take-home message for section 3.13. The nucleic acids, DNA and RNA, are macromolecules that store information in their unique sequences of bases contained in nucleotides. They're building block molecules. Both nucleic acids play central roles in directing protein production in organisms. Moving on, A molecule of DNA has two strands, each a sugar-phosphate backbone with a base sticking out from each sugar molecule. The two strands wrap around each other, turning in a spiral, and the two strands are connected by the bases protruding from them. You can picture a molecule of DNA as a ladder. The two sugar-phosphate backbones are like the long vertical sides of the ladder that give it height. A base sticking out represents a rung on the ladder, or more accurately, half a rung. The bases protruding from each strand meet in the center and bind to each other via hydrogen bonds. DNA differs slightly from a ladder, though, in that it has a gradual twist. The two spiraling strands together are said to form what we call a double helix. Moving on, the two intertwining spirals fit together because only two combinations of bases can pair up together. The base A always pairs with T, and C always pairs with G. Consequently, if the base sequence of one of the spirals is C, 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 T, T, A, G, G, A, C, C, the base sequence of the other must be G, 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 A, T, C, C, T, T, G, G. That is why researchers sequencing part of the human genome describe only one sequence of nucleotides when presenting a DNA sequence, even though that DNA is double-stranded in our bodies. The sequences of nucleotide bases containing the information about how to produce a particular protein have anywhere from 100 to several thousand bases. In humans, all of the DNA in a cell containing all of the instructions for every protein that humans must produce contains about 3 billion base pairs. Almost all of this DNA is in the cell's nucleus. And that brings us to our take-home message for section 3.14, and that is DNA is shaped like a ladder in which the long vertical sides are made from a sequence of sugar-phosphate molecules, and the rungs are pairs of nucleotide bases. The sequence of nucleotide bases contains the information about how to produce a particular protein. Moving on, the process of building a protein from a DNA sequence is not a direct one. Instead, RNA serves the role of an intermediary. First, a copy of the DNA sequence is encoded in RNA, and then that RNA copy is used to direct the production of a polypeptide. It's not surprising, then, that DNA and RNA have some structural similarities. There are, however, three important differences between DNA and RNA molecules. First, the sugar portion of the nucleic acid contains an extra atom of oxygen. Second, while RNA has the bases A, G, and C, it does not have the base T. Instead, it has a similar base called uracil, or we abbreviate that U. Thirdly, RNA is a single-stranded molecule, so its bases do not bind with the bases in another RNA strand to form a ladder-like structure that we see in DNA. So, when a cell needs to synthesize a protein, a short strip of RNA is produced using a segment of a DNA strand as one model. The RNA nucleotides are therefore complementary to the DNA nucleotides, so the RNA molecule contains all the information present in the order of nucleotides in the DNA segment. The RNA moves to another part of the cell and then directs the linking together of amino acids to form a polypeptide chain that folds into a three-dimensional protein. And that brings us to our take-home message for section 3.15, and that is RNA acts as an intermediary, taking the instructions for protein production from DNA to another part of the cell, where, in accordance with the RNA instructions, amino acids are linked together into proteins.